separate sheet, make things easy on yourself so that you have a separate drug list. And then when we get to the end of the class, on the final, you can assume you've got all your drugs there on one piece of paper, and hopefully you don't have to scramble around and try to find them all throughout your notes. So that's kind of the general game plan going forward. I'll continue to talk about that every night. Last Wednesday night, we began to get into a discussion of skeletal muscle physiology. And I showed you an overview of the anatomy of skeletal muscle. That file's posted under course contents. Make sure you look at it. Make sure you have it memorized. And then I began to explain to you the four fundamental physiological processes that basically play the fundamental role in the ability of muscle to do what muscles do. What do muscles do? They contract. And when they contract, they produce forces. And they can produce changes in length. So what are the four processes? Well, hopefully you have it in your notes. The first is excitation. That's an electrophysiological set of events. Then there's excitation, contraction, coupling. That's a transduction series of events. Then there's the contraction phase itself. That's mechanical in nature. And then relaxation is a combination of electromechanical events. Once I'm done describing that, which I will be tonight, then we're going to get into the biochemistry of the so-called cross-bridge cycle. And we'll talk about different types of molecular motors. And I'll present to you the concept of muscle fiber types. I suspect that many of you learned a little bit about muscle fiber types in your anatomy course. If you didn't, you'll get exposed to it tonight. If you did, I'm going to build upon what you learned previously. I'm going to talk to you about the structure of the myosin molecule, which is the molecular motor. Myosin is a molecular motor. And probably that's as far as we'll get tonight. And when we come back next week, then we'll get into the structure, sarcomeres. I'll talk to you about some basic properties of muscle mechanics, and then we'll talk about the concept of motor units and recruitments. So we got a lot coming up, a lot. So if you look at your notes, you guys help me out. When we finished up with excitation, I described for you what? 8 events? Was that the number 8?
Yes.
Okay. And number 8 should have been the depolarization of the sarcolemma. Yeah? No? Maybe so? All right. By the way, can you guys actually see the blue in the back there or not? Good, I found a color of blue that worked. Well, so we've got this connection between the motor neuron and the muscle fiber itself. And this connection is referred to as the neuromuscular junction. And all of you would tell me that the neurotransmitter that's released from the motor neuron is acetylcholine. And acetylcholine binds to a so-called nicotinic ACH receptor. And if you're asked to describe some of the properties of this nicotinic ACH receptor, you might sit there and say it's a ionotropic system. You might sit there and say that it has different subunits. And some of those subunits are the ligand binding sites. And the ligand binding sites are found at, or are referred to as the so-called alpha subunits. And there are 2 of them. And then there's an ion channel. And you're going to know that that ion channel is conductive to both sodium and potassium. So it's what I described for you as being promiscuous, meaning that it allows more than one ion to move through it. And so the question is that if we have sodium ions moving into the cell and we have potassium ions moving out of the cell through this ion channel, how the heck is it that, that the skeletal muscle fiber is able to depolarize? And the answer is the electrochemical gradient for sodium ions is much greater than the electrochemical gradient for potassium ions, and therefore the positive charge moving in versus the positive charge moving out is much greater. That causes the muscle fiber to depolarize. That then causes voltage-gated sodium ion channels on either side of the acetylcholine receptor to open up, and we have the propagation of the action potential down along the length of the sarcolemma in both directions. Remember, one of the points that I made to you is that the neuromuscular junction is approximately in the middle of the muscle fiber. So the action potentials have got to go in both directions to get the muscle fiber to contract properly. All right, that then takes us to the importance of the so-called transverse tubules. So let me just say that the transverse tubule is nothing more than an invagination of the sarcolemma. So listen carefully. Everybody just look up here. The concentration of potassium here is 4 millimolar. The concentration of sodium here is 140 millimolar. The concentration of potassium here is 4 millimolar. The concentration here of sodium is 140 millimolar. The chemical composition of the fluid in the T-tubule is exactly the same as the composition of the fluid outside, because that's what it is. In the T-tubule, the chemical composition of the fluid here is nothing more than the interstitial fluid. So now we're into number 9. And in number 9, we have the depolarization of the T-tubule membrane. Importantly, that then leads to number 10, which is the opening of these voltage-gated calcium ion channels. And the cool thing about this is it reinforces one of the themes that you learned over the course of the last 4 weeks plus, and that is calcium is this incredibly important intracellular signaling ion. And one of the reasons that it can play a role in terms of intracellular signaling is because the concentration of calcium inside the cell under normal resting conditions is incredibly low. So now all of a sudden, concentration of calcium goes up, the cell sees that, and it responds accordingly, depending upon what type of cell it is. In this case, we're talking about a skeletal muscle fiber. So number 10 is the opening of voltage-gated calcium ion channels found within the membrane of the T-tubule. And just to reinforce one of the points I made to you last week is that the distribution of the voltage-gated calcium ion channels is found in the T-tubule, not along the length of the sarcolemma. So up here, there are no voltage-gated calcium ion channels. No voltage-gated calcium ion channels here. They're found here in the membrane of the T-tubule. Well, we've got this organelle. It's a modified version of the endoplasmic reticulum. And the job of this sarcoplasmic reticulum is to control the level of calcium in the sarcoplasm. The sarcoplasm is the cytoplasm of a skeletal muscle fiber. And it does this through two different mechanisms. There are the release channels for calcium. Those are found on the end of the sarcoplasmic reticulum. And hopefully all of you will tell me that this region of the sarcoplasmic reticulum is known as a terminal cisternae. All of you should have that in your notes because I mentioned this to you last week. So the calcium release channels allow calcium to come out. But also I mentioned that there's this other region of the sarcoplasmic reticulum, the membranes, the so-called lateral border. And here we find calcium ATPase pumps. And here's a simple way to think about this. The calcium release channels are the on switch. Really, it's the binding of calcium to tropin C. Again, you learned that last week. And the off switch are the calcium ATPase pumps. So here in parentheses, I'm just going to put off switch. And over here, underneath the calcium release channels, I'm going to put on switch. Also, I mentioned to you last week that there's a physical connection between the voltage-gated calcium ion channels and the calcium release channels. So there's a protein that physically connects these two proteins. And this we're going to refer to as a linker. What happens is that when the membrane of the T-tubule depolarizes, that causes the voltage-gated calcium ion channels to open up. And when they do that, they change their structure. And when they change their structure, they put tension into this linking protein. That then affects or alters the structure of the calcium release channel, allowing calcium to flow out of the sarcoplasmic reticulum according to the electrochemical gradient for calcium. And here, once the calcium is released into the sarcoplasm, calcium will bind to a calcium binding protein. You already know that one type of calcium binding protein is calmodulin, and I mentioned to you that there's a superfamily of calcium binding proteins. There's another member of this superfamily known as tropinin C. That C stands for calcium. And so this is a type of tropinin that has specific binding sites for calcium. Just to emphasize, as I tell you every time, the convention I'm using is when you see a plus between two molecules or ions, that means that they're dissociated, they're not bound to one another, they're simply moving around through diffusive processes. And when you see an asterisk, that is just a notation. It's a convention that I'm using to indicate that something is bound to something else. So in this case, I've got calcium with an asterisk, and then I'm putting capital T, lowercase n, capital C. And you simply read that as calcium is now bound to troponin C. All right, so number 10 is the opening of the voltage-gated calcium ion channels. Number 11 is a conformational change in the linking protein. Number 12 is the opening of the calcium release channels. Number 13. Number 13 is a binding of calcium to troponin C. Now, a key point. I'm not going to test you on this, but these calcium ATPase pumps, the speed at which they're working is dependent upon the levels of calcium in the sarcoplasm. When the levels of calcium in the sarcoplasm are very low, then the pumps are running at a slow speed. When the concentration of calcium in the sarcoplasm goes up, then these run at full speed. How is it that we can get a muscle to contract if we have these two opposing phenomena? We've got the release of calcium, but simultaneously we have the uptake of calcium. How is it that we can actually get a muscle to contract if calcium is a signal that turns on the contractile machinery? And the answer is that the release of calcium is like this huge flood that these pumps cannot keep up with. The pumps don't have the ability to keep up with the rate at which calcium can be released from the sarcoplasmic reticulum. Hence, the release of calcium from the sarcoplasmic reticulum overwhelms the ability of the calcium ATPase pumps. Let me just stop there and ask if you have any questions. Don't be bashful to speak up. Yes, go for it.
Quick question. So when the voltage-gated calcium ion channels open up, that's also letting calcium in.
It is, but it's a relatively small amount compared to the sarcoplasmic reticulum. And I can't get into all the minutiae of it, but what you have to appreciate is that this cell is relatively big. We have thousands upon thousands of sarcomeres in series. And we have to be able to flood each one of them with calcium that quick to get them to all work together. See, that's a big problem here, is that we've got hundreds of millions of these things, these sarcomere, right, last week. We've got hundreds of millions of these in a single cell, and we have to get them synchronized so they're all working at exactly the same time. So we have to have a way in which excitation spreads down along the length of the sarcolemma very, very fast. And it does, because that's an electrical event. That's happening incredibly fast. The mechanical event's slower, 100 times slower. But because we've got thousands of them in parallel, we've got to be able to release. We've got to get these voltage-gated calcium ion channels to open up so that all the sarcoplasmic reticulum are releasing their calcium simultaneously. And around each set of sarcomeres in series, there's a sarcoplasmic reticulum. So there's a sarcoplasmic reticulum around this set of sarcomeres. Here's another set of sarcomeres. These sarcomeres in series are known as myofibrils. So there's a sarcoplasmic reticulum wrapped around this myofibril and a sarcoplasmic reticulum wrapped around that myofibril. because, like I said, we have to drench the system almost instantaneously to get all these guys to work in synchrony. It's an amazing story. What skeletal muscle does is mind-boggling, and as I used to tell the medical students, skeletal muscle is the most amazing cell type in the body. Of course, I'm being somewhat facetious when I say that, but when you actually study the structure of skeletal muscle, it's incredibly complex. And I just had a visit to my doctor, and he had one of the medicals, well, he had a resident in there, and this guy goes, "Dr. Caiazzo, I had you for med physiology." All right, but our story's not done. But before I go there, make sure you understand that these voltage-gated calcium ion channels can be described using a different name, and that name is, these can also be described as dihydropyridine receptors. As I stated to you last week, why are they referred to as dihydropyridine receptors? Because dihydropyridines are a class of drugs that bind to voltage-gated calcium ion channels and close them. They block them. So they're called calcium channel blockers. This will become very important when we start talking about the heart. A very, very popular class of drugs used to treat a variety of issues related to the heart. Our calcium release channels also go by a different name. Sometimes you'll hear the calcium release channels described as ryanodine receptors. And you'll see the abbreviation capital R, lowercase y, capital R. And why are they referred to as ryanodine receptors? Because there's a class of drugs known as ryanodine. And ryanodine specifically binds to these calcium release channels and will block them. So make sure you're familiar with those two different types of names. That's fair game for a test. I'm going to erase this label here. Or just to make sure, we're talking about excitation, contraction, coupling. This is a transduction event. We're not done with that story. Well, if you're listening to what I said a few moments ago, here's a myofibril. By the way, myofibril is nothing more than a bunch of sarcomeres in series with one another. What's wrapped around the myofibril? Nope. The sarcoplasmic reticulum is wrapped around these sarcomeres so that we can instantaneously activate all these sarcomeres together. What's wrapped around this myofibril? Sarcoplasmic reticulum. Here's two myofibrils in parallel with one another. Sarcomeres in parallel determines what mechanical property? Force production. Sarcomeres in series determine what property? Come on. Yes, here we go. Here's a myofibril only made up of one sarcomere. That never happens, but we're going to say it does. And listen carefully. Here's a hypothetical. This can shorten 1/2 of its length. Well, I have two of them in series. This one can shorten half of its length. This one can shorten half of its length. What's the total length that these two sarcomeres can produce? The total length change. Say loud. One sarcomere length, right? Here's a third and a fourth. And now, what's the total length change that this myofibril can produce? And your answer is 2 sarcomere lengths of change, right? So the number of sarcomeres in series determines the length change that a myofibril or a muscle fiber can produce. Here's another sarcoplasmic reticulum. Test question. See how well you're listening. That sarcoplasmic reticulum is wrapped around a what? A myofibril. That's exactly right. So what I'm going to do is I'm going to erase the sarcoplasmic reticulum and show you the structure of a sarcomere. By the way, here's Logic 101. I like this question. Every semester I use it. And every semester I get fun answers. Here's a myofibril right here. Everybody look at me, please. Here's a myofibril. And I got this saran wrap. Saran wrap. All of you know saran wrap, right? Good. You got a saran wrap wrapped around this myofibril. Here's the question. Is my myofibril in the sarcoplasmic reticulum? It is, right? Isn't that Logic 101? Miss Elizabeth, would you agree with me? You don't know? I mean, if I have saran wrap and I wrap it around my arm, isn't my arm inside the saran wrap? Mr. Vargas, you'd say what? Are you going to disagree with me?
I wouldn't say it's in it. I'd say it's wrapped by it.
What would you say? Is my arm inside the saran wrap or not?
No, it's just.
Of course it's not. If my arm was inside that saran wrap, my arm would have to be incredibly thin, right, to be in the saran wrap. Yeah? No? Maybe so? Absolutely. That's Logic 101. That sarcoplasmic reticulum is wrapped around a sarcomere, but the sarcomere is not within the sarcoplasmic reticulum. The sarcoplasmic reticulum is wrapped around it. Everybody with me on this or not? So now I've just erased, magically got rid of our sarcoplasmic reticulum. And now we're going to look at our sarcomere. And I'm just showing one sarcomere, but where I'm putting one sarcomere, you could put 1,000. Now, Mr. Cowell, you had anatomy last semester, right?
Yes, I did.
Yes, you did. Can you describe the structure of a sarcomere for me? You must have learned this in anatomy.
Yes. It starts from, its unit is from Z line to Z line.
Ah, he says its unit is from Z line to Z line. Excellent, good job, Michael. So we're going to say that this represents a Z line. Why are they called Z line? What the hell does Z-line stand for? Tonight, you're going to learn a little German. Zwischen, that means between. When I started high school, meeting with my counselor, Question was, what do you think you want to do? I told him I want to be a scientist. Oh, okay, good. Then you've got to learn to speak German, because back then German was supposedly the scientific language of the world. What is the scientific language of the world, Mr. Corey?
Depends on which science, but I would say either Greek or Latin.
Greek or Latin? Wow.
Foundational laws.
I'm not asking that. I'm asking what's the common scientific language?
Oh, English.
It is English, that's exactly right. So like a fool, I spent 5 years learning German. How many times have I used German over the past 50 years? Muy poquito? Very few times. I did use it this year when we were in Switzerland, and this German guy told me that we were sitting in the wrong seats on the train. He was the conductor, and I had asked him before I got on the train, is this the right train? He goes, yep. So we get on the train, we sit in the seats we're assigned to, and he comes walking up to me in broken language. He goes, you're in the wrong seat, you gotta get off, you're on the wrong train. I'm going, I'm just talking to you. And he keeps talking, and I finally said to him in German, I go, ich nicht verstehen Sie, which means I don't understand what you're saying. But that's about as much German as I've used. Anyways, we see these terms, they have an old historical context to them. You hear EKG versus ECG. Electrocardiogram, spelled with a K, that's German. That's going to be on the test. Mr. Cow, so we've got 2 Z lines at both ends of the sarcomere. What's in between? See, I'm asking these questions because his answers on the test are exceptionally good. And I can tell he goes way, way, way beyond what we talked about in this class.
So in between will be the H zone.
There's an H zone, okay. But there's filaments, right?
And we're talking about A band and I band.
But there's filaments, right?
Thick filament, thin filament.
There we go. There are thick filaments and thin filaments. That was good. I'm going to put little circular-like structures or drawings up here. And each one of these green circles represents a protein, an individual protein or molecule known as actin. And these actin, individual actin monomers, these individual molecules of actin are stuck one end onto the other to form what is called the actin filament. So what I've drawn up here, you'll simply refer to as the actin filament. And here's an amazing part of this story is that each one of these actin molecules has a sequence of amino acids that the molecular motor binds to. We're going to call that the myosin binding site. So for each of these circles, I'm just going to put a little black dot . And just to repeat what I said, Each one of these black dots represents a sequence of amino acids within a given actin molecule that is the binding site for the molecular motor. And we're going to refer to these as myosin binding sites. The molecular motor is known as myosin. To be clear, this is not myosin. These are a sequence of amino acids within each actin molecule to which myosin will bind. Next part you got to pay very, very careful attention to is that, and I'm not going to show this for each of these, but wrapped around the actin filament is a protein, another protein. And if you take a look at what I've done, is I'm showing this other protein being wrapped around the actin filament and covering up, hiding the myosin binding sites. So I'm trying to represent that by simply showing the line of this protein going over these myosin binding sites. And this protein that's wrapped around the actin filament is known as tropomyosin. So again, just to restate myself, wrapped around the actin filament is a protein that covers up, it hides the myosin binding sites. And bound to this molecule known as tropomyosin, there are 3 other proteins. The one in black is known as troponin T. Why is it referred to as troponin T? The T tells us that it's bound to tropomyosin. Mr. Cow,
Yes.
what are the other types of troponin? Do you know?
Is one that bind to calcium,
Okay, what's it called?
troponin C.
That's
perhaps?
correct. That is correct. And what's another one?
And the other one is binding to actin.
Well, we got troponin. No, there's none binding to actin. Okay, so the one I'm showing here in blue, this is troponin I. And the one I'm showing in green is troponin C. So let's just back up for a moment, if I can have all eyes up here. Calcium is released from the sarcoplasmic reticulum. What I wrote up here is that the calcium flows out of the sarcoplasmic reticulum according to the electrochemical gradient for calcium, and it binds to troponin C. Well, that's not really happening here, right? It's happening over here. The calcium is binding to troponin C. Now, what does that I stand for? Say it louder.
Insulin? Insulin.
No. Inhibitory. Stands for inhibitory. So the I, inhibitory. What are we inhibiting? We're inhibiting the ability of the molecular motor to bind to the actin filament. So what I drew up top, I'm going to draw on the bottom. Don't know how well you can see it in the back there. But up top, I'm showing the tropomyosin so that it's covering up these myosin binding sites. Here, I'm showing that the structure of the tropomyosin molecule has changed, and now it's rotated around the actin filament. In so doing, it's uncovered the myosin binding sites. Here I'm going to show the presence of troponin T using that black circle. I'm going to show the presence of troponin I with a blue circle. And I'm going to show the presence of troponin C with a green circle. Except what I'm going to do is where I've got troponin C, I'm going to put an asterisk. And next to that asterisk, I'm going to put the following initials: capital C, lowercase a++. So when calcium is released from the sarcoplasmic reticulum, it binds to troponin C. Troponin C then acts on tropomyosin. So here's a simple example. Just if you watch me, please, if everybody look at me. My arms are tropomyosin. In between my arms is an actin filament. And right now, my hand is covering up the myosin binding site. Attached to my arms, this is tropomyosin, is troponin T, troponin I, and troponin C. In the absence of calcium, troponin I exerts the dominant effect on the structure of tropomyosin, such that tropomyosin is hiding, it's blocking the myosin binding sites. Now, calcium is released by the sarcoplasmic reticulum. I've got troponin T, troponin I, troponin C here. Calcium binds to troponin C, and what happens is my arm rotates like this radially. And when it does that, it uncovers the myosin binding sites. Everybody with me on this or not? So what's happening is the tropomyosin is wrapped around the actin filament, and it simply is rotating around the longitudinal axis of that filament, depending upon whether calcium is present or if it's absent. If calcium is absent, troponin I exerts the dominant effect, causing the tropomyosin to hide or to block all the myosin binding sites. If calcium is bound to troponin C, then troponin C is exerting the dominant effect on tropomyosin, causing the tropomyosin to rotate and uncover the myosin binding sites. Now, I keep saying this, and I hope that all of you will come away from here being better students of biology. But I told you the very first night, life is a miracle. sit there and think about the complexity of what I'm describing for you. How the heck did all this come to be? And we're not even thinking about cells talking to cells and figuring out how to talk to people, how people can take pressure waves and somehow decipher what I'm saying. I mean, it's all magical. It's all really quite amazing. And all of you, as you go out in the clinical world, as I will say to you, and I've already said, is that you're going to be treating other miracles. Those miracles demand, they need to be treated with the best, given the best possible care that you can provide. Never, ever short your patients. Okay, now, Michael properly told us that there are so-called thick and thin filaments. What is a thin filament? Well, there's an actin filament. The actin filament is simply actin monomers stuck one end to the other. That's the actin filament. Please listen to what I'm saying. But the thin filament is the actin filament. The thin filament is here. Here. The thin filament is the actin filament plus tropomyosin plus troponin C plus troponin I. So back up, troponin T, troponin I, troponin C. That's the thin filament. So if I ask you what makes an actin filament, you're going to just sit there and say, just a bunch of actin monomers stuck one into the other. If I ask you what makes up a thin filament, you're going to sit there and say the actin filament, tropomyosin, troponin T, troponin I, troponin C. Everybody with me on that? Well, in the middle, there's a so-called thick filament. And this is where we find our molecular motors. And there are these little projections. Coming off the thick filament. And these little projections are part of the myosin molecule and are going to represent what we call cross bridges. So if you can imagine a gap between the thick filament and the thin filament, when the binding sites are exposed, the heads of these myosin molecules are going to attach. They're going to bridge that gap. Cross bridges. So I'm going to come along. I'm just going to put a little dotted box. Oops. Around this part here. That's our thin filament. And then we've got here in red the thick filament. What's the main protein that makes up the thick filament? Myosin. What is myosin? It's a molecular motor. Mr. Lamb, you there with me? The myosin molecule is going to attach to a myosin binding site. And it's going to go through, it's going to move in this direction. Are you following me on that? So the head of the myosin molecule attaches to this myosin binding site, and then it goes, it moves in this direction. So I'm just going to show a arc with an arrow at the end. And I'm going to do the same thing on the opposite side. And so as this myosin molecule attaches and goes through its power stroke, this direction, what's going to happen to the length of the sarcomere? Yeah, that's exactly right. It's going to shorten. That's exactly right. Does that make sense to everybody? Yeah? No? Maybe so? So imagine the calcium is released. Our molecular motors, look at what I'm doing up here. There's our two molecular motors. There's a whole bunch more. I'm just showing two. And they go through this cycle and move inward. In doing that, they're causing the ends of the sarcomere, which are the Z lines, to move inward. And therefore, you would sit there and say that the sarcomere is going to shorten, right? What do you think? Make sense? That makes sense. Confused? Speak up if you're confused, please. So here I'm gonna, where I've got one of these arcs with the arrow, I'm just gonna put power stroke. Okay, so now let's step backwards. We left off at 13, calcium bound to troponin C. Step 14. Step 14 is the rotation of tropomyosin that then uncovers the myosin binding sites. So in this box, excitation, contraction, coupling, we're starting off with step number 9. Step number 9, as we've already described, is the depolarization of the transverse tubule membrane. Number 10. That then leads to the opening of the voltage-gated calcium ion channels in the T-tubule. Number 11. Just making sure I got corresponding numbers up there. Number 11 is strain of linker protein. Number 12 should be the opening of our calcium release channels. Number 13. The binding of calcium to troponin C. Number 14. Conformational Conformational, what does that mean?
Oh, change shape, structure.
Thank you. Structural, right? Conformational change, structural change in tropomyosin. Tropomyosin, shorthand or abbreviations, capital T, lowercase m. And I'm going to put here as part of 14. So conformational change of tropomyosin and the uncovering of myosin binding sites. Again, if there are any questions, please raise your hand. I want to erase what I've written over there. Are you guys good with that? Everybody's got that down?
No.
For your hearing.
Okay.
All right, now we're going to talk about the contractile process. So now what I'm going to do is that, just to repeat myself, to be clear, is that these little projections off here are individual myosin molecules. Myosin is a molecular motor. You can almost look at that, at least when I look at it, I can envision a rowing boat. And there are these oars coming off. And when I put those oars in the water and I pull on them, they kind of propel the boat forward. And so we've got these little oar-like structures, and they go through a power stroke. When I pull on those oars, I'm producing a power stroke, and it powers the boat, causes it to move. In this case, when we attach to actin and we go through our power stroke, we're putting tension, we're creating force, and we're causing these Z lines to come together. Now, I call this a molecular motor. Why do I call it that? Well, because as it turns out, each one of these cross-bridge cycles produces a piconewton of force. Ooh, piconewton. Go take a look at that metric system and you're going to find out a pico is a really, really, really small thing. So if each one of those myosin molecules is creating a piconewton, a pico-pound of force, how the heck is it that I can curl 150 pounds with my biceps brachii? Well, maybe I can't curl 150 pounds. Maybe I can get up to 60 though. How can I do that? How can that muscle produce so damn much force? And there's one obvious answer. If each one of those is creating an incredibly small amount of force, then how many of those molecular motors are there in a muscle? And your answer is gazillions. Huge, huge number of them. So what I'm going to do is I'm going to describe for you the biochemical steps that occur as this myosin molecule binds to actin, goes through its power stroke, then resets itself so that it can go through another power stroke, and it just simply is oscillating back and forth, producing force, then resetting itself, producing force, resetting itself, producing force. And literally, no joke, when I do this, when I sit there and take my biceps brachiac and do that, there were 10 to the 15th numbers of those events that just took place. That's a number that's far, far bigger than any of us can count in the course of our lifetime. So this story, I'm going to describe for you the biochemistry of the cross-bridge cycle. What is the cross bridge? The cross bridge is the head of a myosin molecule. And I'll just say that over the course of my time, this is just a tip of the hat to the mentors I had. When I was at UCLA, I had the opportunity to learn from the best in the world. People were studying the thermodynamics of these processes. And, you know, talk about being in an environment where you just feel like, man, you are learning so much on a daily basis. It's going to happen to you guys when you get out there into your clinical realm and start learning about all these important things. You'll just sit there and feed off of all that information. Okay, so I'm going to use some initials in telling this story. When I use a capital M, that means myosin. So in the story I'm going to describe, capital M is myosin. That's our molecular motor. Capital A is actin. Remember, myosin is binding to actin, and when it does that, it goes through its power stroke and produces force. So here's how our story starts out. It starts out, we have a muscle at rest. And we have actin plus, meaning dissociated, everything's separate from one another, plus myosin. And bound to myosin is ADP. And phosphate. Kiara. Is Kiara here? No, Kiara. All right. Kent, where's Kent? Kent. So we've got actin plus myosin, bound to myosin, or ADP, and phosphate. Is this attached state or detached state?
Detached.
Thumbs up. That's exactly right. How does he know that? Because he sees that plus sign between the actin and the myosin. So beneath this, in parentheses, I'm going to write the word detached. Well, we have our neurotransmitter that's released, that then binds to the nicotinic ACH receptor at the neuromuscular junction. That leads to depolarization of the sarcolemma, depolarization of the membrane and the transverse tubule, the opening of voltage gated calcium ion channels, release of calcium. And so up above here, I'm going to write release of calcium. And now I'm going to rewrite my statement that I have over here on the left. and I'm gonna put actin. And now, when calcium binds to troponin C, that causes the tropomyosin to rotate. That exposes the myosin binding sites. And now, the head of the myosin molecule attaches to that. So here I've got actin, and bound to the actin is myosin. Bound to the myosin are ADP and phosphate. Kim, you can tell me that's attached or detached? It's attached. That's right here with the asterisk. Okay, the next step in the cross-bridge cycle. We have actin. Bound to actin is myosin. Bound to the myosin is ADP. At this step, phosphate is released. Chloe, where's Chloe? Chloe, one of the things I've talked about all semester is that phosphorylation and dephosphorylation do what? Two thumbs up, very good, excellent, that's exactly right, is that a lot of what happens in our cells is dependent upon phosphorylation or dephosphorylation. And here, this is a dephosphorylation phenomena. And if you can recall what I said earlier, here's our myosin molecule. Calcium binds to troponin C. Tropomyosin rotates around the actin filament, uncovering the myosin binding site. The myosin binds to the actin, and then it goes through this, what did I call this event? Where it's producing force.
Stroke? Stroke?
The power stroke, right? This is the power stroke, is that we've got this dephosphorylation, which is changing the structure of our myosin molecule, causing it to go through its power stroke. I'm not going to put it in here, but I need you to put it in your notes. During that power stroke, what is happening? The answer is force production and length change. Force production, length change. Next step in this so-called cross-bridge cycle. Is that now we have actin, and bound to the actin is myosin. And now we've got the release of ADP. Next step. we have actin plus myosin, and bound to the myosin is ATP. So here, the myosin is detached from the actin. What caused the myosin to detach from the actin? Miss you, Chloe. There is a logic or a reason why I came back to you for this question, is there not? That's what's happened, right, as we've come along, and now we're phosphorylating this molecule and we're changing its structure. And it's the binding of ATP to myosin that causes the myosin to lose its affinity for actin. You guys all wrote that down, right, as I said that. It's the binding of ATP that causes the myosin to lose its affinity for actin. One last time. It's the binding of ATP to myosin that causes the myosin to lose its affinity for actin. And that causes it to detach. Stay with me now. Everybody look up here. Here's our myosin molecule. It's in a detached state. We release calcium. Calcium binds to tropin C. That causes tropomyosin to change its structure and rotate along the longitudinal axis of the actin filament. That uncovers the myosin binding sites. Now the myosin is going to bind. It's attached. Once it attaches, we release the phosphate and the myosin molecule goes through its power stroke. Now we're here. Now we're here. We come up here and the myosin loses its affinity for the actin and detaches. But look at the position I'm showing that had the myosin molecule in. We got to get it back to where it started. Let me see here. Let's see if I have somebody here. Yes. Is there anybody here with the name of Chloe? She's an expert on phosphorylation and dephosphorylation. And she's going to tell us that when ATP binds to myosin, it's said to be hydrolyzed. What that really means is we just take the molecule of ATP and we break it apart into ADP and phosphate. And what that does is when that phosphate breaks apart, Mr. Cow, when that phosphate, no, Mr. Curry, when that third phosphate, that bond between the second and the third phosphate is broken, what is released? I would say energy too, right? Energy. And so some of that energy, most of that energy comes off in the form of heat. That's exactly right. For every molecule of ATP that we hydrolyze, we break apart, 75% shows up in the form of heat. But for muscle, the other 25% shows up in terms of doing work. So we come along, we hydrolyze this molecule of ATP, and what that does is it re-energizes the head of our myosin molecule so that it can go through another power stroke. So this event right here, this transition from here to here, that leads to the re-energization of the myosin molecule so that it can go through another power stroke. This is the cross bridge cycle. So we're starting off with event number what? 15 is it? 14 was the movement of tropomyosin and covering the myosin binding sites. Yeah, no, maybe so. Number 15. Where's number 15? Here it is, right here. Here's number 16. Number 17. Number 18. Number 19. Number 20. Number 21. Number 22. Number 23. Okay, listen carefully, please. Listen carefully. So we come along, calcium's released, the myosin binds to the actin, it goes through its power stroke. And then we're going to come along, we're going to re-energize the head of the myosin molecule, and as long as calcium is present, then that same myosin molecule is going to go through another cross-bridge cycle. Another molecule of ATP will be consumed. That will re-energize the head of the myosin molecule. As long as calcium is present, it's just going to keep going through this cycle over and over and over again. As long as calcium is present. Mr. Cow, there are two things that are required to keep that cross-bridge cycle going. I just told you one of them. What's the other one?
The availability of ATP.
Fantastic. Very good. That's exactly right. And the concentration of ATP inside cells is really high, right? Haven't I told you that? Don't think so. I've told you just the opposite. I've told you that you would sit there and think that our cells, because ATP is so darn important, you'd think that they have this plethora of ATP molecules floating around. No. Literally, if nothing else happens, if I don't do anything to resynthesize ATP, that I just used up all my ATP in my muscle. There are other things going on, but if I got rid of those, that's how little ATP there is. A couple contractions, boom, I've exhausted the ATP. All right, now we know that Mr. Cow had anatomy last semester. Who in here is brave enough to sit there and say that they had anatomy also last semester? Ooh, oh wait a minute, what do I hear? Do you guys hear that? Nobody else in here says that they had anatomy last semester. Oh, it's amazingly quiet. Michael, can I ask you a personal question?
Yes.
Did you work with anybody in this room here during your anatomy class last semester?
A few.
A few. Are there any of them sitting close to you?
Yes.
There is. Or there, is that person who's close to you, would they be to the left or the right of you?
This is very sensitive information.
You must have learned that sometimes when our end comes, that the muscles take on this interesting state. And what was it called? Oh, it is called rigor, right? Where's rigor over there?
not sure. I don't know if it's the power shift or if it's 2021.
Well, what do you have to keep that cycle going? Mr. Cow told us. We need two things to keep that cycle going. What are they?
ATP and calcium.
Okay. Now, I reached my last moment of life and I transition off into the great cosmos. I'm now going to see Q from Star Trek. But my muscles take on this stiffness. And that stiffness, is that due to absence of calcium or is it due to absence of something else? Because when I pass on, what happens to my breathing? It does stop. What happens to the oxygen in my blood? What happens, did you learn something about how oxygen is used?
In the electron transport chain.
There we go, that's exactly right. So the electron transport chain stops. And the production of ATP, and where in that cycle does that happen? Where do we need ATP?
For cell phone to re-energize.
We need ATP here, right? So in the absence of ATP, we get stuck where? That's exactly right. And so what do you want to call step 21? We're going to call it the rigor state. That's exactly right. There's your explanation as to why sometimes you'll hear people refer to a dead person as a stiff. Why are they doing that? Because their muscles are in rigor and therefore you can't easily move their limbs and cause the joints to go through flexion and extension. Now that doesn't happen forever, but our muscles soon after our death move on into the rigor state. Okay, sorry I went over forth, but a fun topic to think about. Now you actually know the biology behind rigor. Okay, I'll see you guys upstairs in about, let's say, at 7:10. If you have questions, comments, come on down. More than happy to